So next talk is also by someone I know well, Professor Karthik Raman, who is my colleague in Department of Biotechnology at IIT Madras, and also colleague in many centers together. Karthik works in computational systems biology, and I don't know today what he's going to talk about community models. I have not seen this paper. Correct?
Yeah.
Yeah.
So thanks, Arijit, for inviting me frequently to IMSC, which meant that I had to find out something new to talk about. I can't keep recycling my talks. So this is something absolutely hot off the oven. It's not even off the press yet. So it's only on bioarchive, like early today morning. So the idea here is we want to try and see how we can build community models and try to surmount the uncertainty that arises typically out of using 16S amplicon sequencing data and so on. So just a quick overview about my group, because I'm not giving a nice overview talk that I usually give, and I'm just focusing on a very single study. So this is a broader picture of what we do. We've looked at a lot of approaches for in silico metabolic engineering, synthetic lethals in metabolic networks, systems theoretic approaches to design regulatory networks. But over the last few years, we've been obsessing with microbiome systems biology. We've been looking at various kinds of methods, both constraint-based methods as well as graph-based methods to study interactions between microbes. And we use a bunch of techniques like constraint-based modeling, thanks to Anu for all the introduction, so I don't have to go through anything now. Machine learning, network biology, and a bit of high-performance computing as well. We also work on integrating multi-omic data and work very closely with the Center for Integrative Biology and Systems Medicine and the Robert Bosch Center for Data Science and AI. So these are a lot of things that we do, and our goal is trying to have some impact in the clinic and or the environment. Some of our contributions, so if you see, we've contributed both algorithms in theory as well as data analysis and applications in microbiome and metabolic engineering. So this is broadly the spectrum of stuff that we work on. And just a few more ongoing projects if you want to have something to chat about with my students over tea. So we are trying to look at rare microbiomes in clean rooms using comparative genomics and metagenomics. Also looking at designing microbial communities for bioprocessing, metabolic modeling approaches to identify prebiotics that can modulate growth in communities, and a few other things. We're also looking at the metagenomic map of the Chennai microbiome and things like that. So lots of interesting stuff going on, but all this is for another day. Today I'm going to talk to you about the one I was telling you, mainly in terms of building community models using just 16S data and so on. I don't think I really need to introduce the microbiome. We all have by now been convinced that it's such a huge factor even in cancer and so many other diseases. But typically microorganisms thrive in multi-species and dynamic environments where the community structure is dictated by a complex web of interactions where metabolic interactions typically predominate. So this is why we often obsess with looking at these cells through a metabolic lens, and it does give us a lot of information. And there are many microbiomes of interest. So you see all these interesting microbiomes that are part of EBI's project and so on. So like soil, gut, skin, coral, and whatnot. And these microbial consortia are also useful in applications, industrial applications like in metabolic engineering and so on. But no matter what the application is, it's very important to look at the interactions between these microbes. Okay. And if you see how typical characterization, traditional characterization involves growing some stuff in the lab, but a lot of times you cannot grow these stuff in the lab, so you sequence them and you try to understand what kind of organisms are present and so on. And it's cheaper to do like 16S amplicon sequencing rather than metagenomic or shotgun sequencing, right? So if you see the literature is replete with 16S data sets. And the motivation is that there's a huge number of data sets that are present from 16S amplicon sequencing. It's obviously way more cost effective. But the catch is that 16S only resolves taxa down to family or genus level and almost never to the species level. But if you want to build models like the kind that were being discussed in the morning and earlier yesterday as well by Nirav and so on, you need to know whole genome sequences, or you need to know what are all the enzymes that are present in the metabolic network of an organism. So without this information, how do you try to build a model? So what are genome scale metabolic networks? I think you have a very good introduction from today morning, but just quickly to recap, they've been reconstructed for lots of organisms, essentially a long list of what are all the reactions happening within the cell. And you can build these draft reconstructions very easily from very nice databases. So if you see some of Amit's work, they've curated lots of organisms by starting off with this model seed kind of models, but then building and gap filling and doing a lot of manual curation on top of that. And there are many methods to analyze these models. And ultimately for us, this is what it looks like. It's a network. You have some nodes and edges and things like that. But we also look at the stoichiometry and things like that. So what can genome scale metabolic models tell us? I really like this picture simply because it's quite old now, but it talks about 250 studies using just the E. coli model. And using just the E. coli model, people have looked at a whole host of things, right from metabolic engineering to model-driven discovery, analysis of biological network properties, and so on. And also interspecies interactions, which was then not very popular, but today I think it's taking a life of its own, and there are lots of studies that are looking at interspecies interactions, modeling them using various kinds of modeling approaches and so on. And there are many ways to model these microbial communities. You can use graph-based methods, as we've done a few times, and also constraint-based methods, and population-based methods, agent-based methods, and so on. And in all of these, it's very interesting to look at what kind of metabolites are being exchanged. Is propionate, is butyrate being exchanged, or is propionate being exchanged, and so short-chain fatty acids and things like that. And there are many applications in understanding communities such as the gut and so on. So there's constraint-based modeling, as Anu was mentioning today. So very popular for applications such as metabolic engineering particularly, and involves predictions of growth rates based on certain assumptions about the media conditions and steady state and so on. And it demands well-curated models. There are network-based methods which we've also contributed quite a bit in the recent years, essentially focused on pathfinding in metabolic networks and metabolic exchanges and so on. But in all cases, you need methods that are scalable, simply because we are talking about tens of organisms or sometimes even hundreds of organisms, and of course that they are accurate, and but we also need methods that can handle compositional uncertainty or can make predictions despite the compositional uncertainty. So so some of the challenges here are, although constraint-based models are very useful, they are specific to the species and strains built from whole genome sequences, and a lot of data we have is 16S. So how do we sort of do this bridging? I need whole genome sequences, but I don't have them. Can we work something in between? So my question is, can we build genus-specific metabolic models? To what extent can we do that? And yet mitigate the uncertainty in inferring metabolic capabilities to the extent possible. So this is the question we set out to answer. Are there methods that do it? Yes, there are a couple of methods. There is this method called pan-genome. There are ways to build these pan-genomes from metabolic networks, but typically they just involve building lumped models. So you just take all the known models in a genus, like let's say Bifidobacterium, I know Adolescentis, I know Bifidum, whatever, and so on. I take like 20, 30 of these models and just lump them together, put a common biomass objective function or something like that, and see what are its metabolic capabilities. So it's a very straightforward kind of model. But there is no existing tool that has addressed the complexities in exploiting both the composition data from 16S sequencing and allowing for the variation, right? So you may know some species may be in a data set may be resolved up to the species level, some components, and some may be resolved only to a genus level. So can you handle this kind of hybrid data as well? So these are all the questions we wanted to answer. And Indu is in the audience, so I'm going to redirect half the questions to her. This is her method. So pan-genus model to counter uncertainties in composition and making predictions. Himanshu is also my co-author in this study. So Indu's called it the Panera algorithm. So you can go and look it up on GitHub as well. So what do? We construct what is known as a pan-genus metabolic model using reasonably well curated genome scale metabolic models from this resource called Agora, and some of you who work in the field may have heard of it. It's a huge repository of gut-based microorganisms. The first version had about 800 odd, and the second version had about 7,000 odd organisms that are reasonably well curated, and now they have a new Apollo which has apparently 250,000 organisms, but I haven't looked at it very closely. It's just a week old or so. So we try to build species-aware pan-genus models to understand the capabilities of a given genus and include as much information from amplicon sequencing as possible to figure out what is it this community can really do in practice. Okay, so what are the key steps? So this is a pipeline. The pipeline has three main steps. First thing is a combined model of all reactions, basically pool all the reactions together and formulate a biomass objective function that is some sort of weighted version of all the individual biomass compositions and parameters that accommodate the variation in species composition. So you may or may not have certain species in a community or in a genus. So let's say you have a data set that says that bifidobacterium is present in the gut. And you do not know if bifidum is present or not and things like that, or adolescentis is present or not and so on. So how do you handle this uncertainty? Sometimes you may have, if you have prior knowledge, you can incorporate it. If you have actual accurate knowledge from a good data set, you can incorporate it. If not, how do we handle it? So these are the questions that we were confronted with. So we customize this, so this is just the biomass function which is just weighted by probabilities, and to customize the pan-genus model according to species presence, we also have a species probability vector and we connect this to a reaction species matrix, right? So which species has which reaction and does it ultimately figure into the model or not. So this is in some sense like these context specific models that people generate, but it's at a a little bit more complex level because we are looking at multiple species and their presence, absence and so on. So some questions here, and hopefully we've answered all of these in the affirmative at the end of the study. So can these pan-genus models help explore the metabolic activities of a genus as a whole? And does the metabolic potential vary with the species proportion in a genus? Right, so when you have more of bifidum versus more of adolescentis, how does bifidobacterium metabolism change? Does everything change or do some things remain constant? These are the kind of questions we wanted to answer. And can we employ both ambiguous and unambiguous taxonomic data to study and predict the functional capabilities of microbial communities? Okay. So what do? We start off with a species probability vector. You can say everything is present in uniform probability, or some are present, some are not present, and we also did like random scans and things like that. Customize these pan-genus models based on these probabilities, perform a flux variability analysis which tells you what is the span of observable fluxes under a given set of conditions, and evaluate the flux range and average flux from the maximum flux values, and look at the bandwidth, right? So the bandwidth could be very tight, it could be highly varying. I'll show you even more variation when I go on to the real data, or not so much variation, so low variation in metabolite production. So this is the kind of thing that we wanted to end up predicting by ultimately plugging in this species probability vector. So this species probability will change based on your data set, based on the resolution of your data set, and so on. And these are some very interesting things. I'll first go on over the left part and then go over the right part, because this is a question that a lot of people working with microbiomes typically have. So what you see here is something that's like tightly regulated. So you say that acetate flux is very tightly regulated on both EU diet and Mediterranean diet. So these are two different inputs that is given to the gut flora. This has low variation. It's still in like one corner of the x-axis, but here it's bimodal and it sort of spreads across a lot more. There's a lot of variation in some of these fluxes. So this is what you see in different fluxes. So acetate, in this case, varies quite a lot, whereas in this particular example varies very less. What you see here is that you see these genera that are colored the same. It's difficult to read it. I think it's Prevotella and I don't remember what's the other one. So what you have is these two cluster very close to each other based on their functional or flux profiles. Phylogenetically, they are super distant from each other. So which is why you often find these data sets or even human guts which have very different compositions but very similar phenotypic output or whatever, right? So meaning they have similar health. So two people with diabetes may have very different gut microbiomes, but it's possible that their metabolic profile is fairly similar and so on. So this helps us ask these questions. I don't think we've answered this yet in one way or the other, but we do see that some very different genera can actually cluster very close to each other depending upon their metabolic output rather than just their sequences. So we looked at some synthetic data sets and so on. We created 10 and 50 member data sets and also combined it with available metagenomics and metabolomics data sets. We used what is known as the microbiome modeling toolbox, which has the one existing method for building it's called pan model. It's for building these communities. We used a regular community that's based on these GSMMs. Then we also used pan models from the microbiome modeling toolbox and our pan-genus metabolic model and hybrid models. And we looked at all of these and how do they perform. And it turns out that in some cases there is no difference between any of these, but there are a few cases where the PGMM and the hybrid models can really outperform what you see with the other approaches, right? So we looked at a synthetic data set with community size of just 10 and 50, and you see that in some of the larger data sets the performance of, so the x-axis is, this is a qualitative variation. It just tells you which reactions are present and not present and so on. And if you see, we also did a quantitative variation where you see the PGMM does way better than the pan model, although the hybrid model is not very different from either of the pan or this. Whereas in the 50 member community, you see that the hybrid model has much less error in capturing the reality, the ground truth, which is based on using those GSMM models. Basically accurate community level modeling using the conventional GSMM communities. And here again is based on the metabolites, and you see that the hybrid model has a much better performance in predicting what kind of metabolites are produced and things like that. So this is based on the absolute flux differences. This was based on just a Boolean, whether it is produced or not, and so on. So essentially the pan-genus models can predict the functionalities of communities much better than pan models. And you typically have at least a 10% reduction in prediction error and a lot of flexibility to incorporate various kinds of data sets. You just don't put a one size fits all kind of, all species are present in this genus and so on. That's the kind of vanilla assumption that the pan model goes with. So there are some limitations as well, and a lot more because we are looking at not very well curated models, right? So we rely on these databases such as Agora. We really haven't invested any effort into curating these models. At some point, I think we should. But we are reliant on our resources such as these, but these resources are continually being updated and improved. And I think there are lots of new machine learning tools again which can play a good role in further enhancing these models and so on. But a big catch although is that you need consistent annotations. You need those models that we are putting together to be able to talk to one another. So typically we find that one group calls glucose as GLU, somebody else calls it as GLC, and it's all over the place. So you really can't align the annotations and map these models, although it's far less of a problem today than it was, say, a decade ago. And we used about 7,000 curated models, and this is actually a fairly good resource. So I think it's not such a big limitation right now. So to summarize, I hope I'm doing well for time. So there are not enough methods that can exploit the widespread 16S data that exists, right? There's so much of 16S data. It's so much cheaper, so much easier to generate, and we have historical boatloads of 16S data. And existing methods just lump these to make pan models. They don't give you a way to vary the abundance of various microbes and things like that. So our goal was to minimize the uncertainties while constructing and analyzing these communities from 16S data. And hopefully the hybrid models that we have which seem to really outperform the conventional GSM based communities, can be really useful going forward, because a lot of time we want to make, as a modeler, I don't want to complain about the lack of data. I want to deal with the car. I want to play with the cards I've been dealt with, and see what is the best you can make. What is the best use you can make of given a bunch of data sets. So thanks to Indu for doing all the work. She's in the audience. You can catch her a little later, and also to Himanshu and all our funders. So with that, I'm happy to take questions. We just put this on bioarchive earlier in the day, so please feel free to read it and share your comments before we send it out for review. Thank you.
Thank you, Karthik. We have time for a couple of questions. Okay, I'll start. Okay.
Very nice work, Karthik. So you also showed that the metabolic profiles could have its own measure and bring together apparently diverse microbiota together in a phylogeny. But I wonder whether the extent of the microbiota present in the gut matters at all, because it can also be quite dynamic.
True. So there are lots of studies here, and the jury is completely out on this. But there's a classic study which talks about 3 different enterotypes. So there are 3 common groups of, 3 sets of microbes, non-overlapping to an extent. I think some species are common, but again, there's very little done on the Indian gut microbiome. So, and that project is almost done now. It is being headed by Yogesh Chauche from, he was at NCCS, right, in Pune. And I think when we get the data from that, we do see a lot of Prevotella presence, which is not common in the other data sets and so on. So lots to be understood. But to your point, I think there is a lot of understanding that despite the variation, there are similarities in the gut profile. If you look at the SCA, the short chain fatty acid profile and so on, there is similarity across certain conditions. But depending upon how you look at it, it really seems to spread. So Indu has done quite a bit of analysis. We've been looking at infantile diarrhea and so on. And what we see that sometimes if you project the data, you see that the case and control have a substantial amount of overlap. Okay, so people with disease and people without disease can have very similar microbiomes. But in some cases, there is enough signal there as well. So it still remains to be teased out. So I don't know if I answered your question, but essentially we are searching for an answer still.
Thank you.
I have a question regarding the modeling framework itself. So when you are considering these whole genome metabolic models, is there an advantage to be gained if you coarse grain the model to some extent to say that now instead of looking at all the features, I look at some abstraction of these things and of say molecules and just focus on the main ones and then?
I think that's a good point. We haven't tried it essentially because we didn't run into any computational challenges or things like that. But going back to your point, I think that's a very good idea. Why worry about glycolysis if it's present everywhere? Just reduce it to like a single reaction of glucose giving pyruvate, right? It's not going to really, may not be impacting, but we don't know. And currently it's very easy to build these models and simulate them. So and without really if we were struggling with the computational time or something like that, I think that's where we would look at. But currently it's not an issue. But if we started really building large communities, maybe it's worth doing that. I think Chetan has a question.
Sorry, I have sort of continuing on the question. So there are 2 questions, which is one, given that the map for individual strains is incomplete, why would you think that a map, that adds many wrong models will give a better answer? And actually, my question stems from ignorance. And what is the gold standard? So when you said that one model outperforms the other, and the last question is that you have put the objective function as a weighted sum of individuals, but will not each species try to maximize? Will it not have a weightage of 1? You understand? Will not.
Yeah. So I think all of these are possible. So your question is very valid in terms of why put multiple wrong things together and see what comes. So the question we started out answering was that even if you put these, are there certain things that are conserved? No matter whether the bifidobacterium is 0 or 100, I still get the same amount of acetate, at least in certain conditions. So that is the question we started off, and it looks like there are enough fluxes or enough things that seem to have low bands of uncertainty despite so much uncertainty in the input. And that is the interesting part. But otherwise your point is obviously valid, and there are of course, the better data we get, the tighter the predictions will become. Are there any tight predictions with super loose data? So this is sort of the question that the gold standard is using the classic community modeling approaches, which take all of these where you know what species are present and so on. So we take so we use synthetic data sets where we know the ground truth. We know X of this is present, Y of this is present, and so there are ways to model that. So that versus now I make it uncertain, right? I drop this information and basically confound it and pass it to my modeling system, and is it still able to make at least a few tight predictions and so on? Yeah, that's one more question. Arijit, the next.
Okay, so I can ask at the end. That's okay.
My question also stems out of ignorance. Let's say if you take one model separately and simulate it, right? It's anyway going to take the exchanges are going to be for all the metabolites, and I'm assuming that your the metabolites are abundantly available for all the different networks in the community model also. So why is a separate simulation different from a community-based simulation for a particular bacterial species?
True. I think that's a good point, but the thing is, when you put all these together, right, the ability of one organism, there are some organisms that can use the food that's available better, depending upon what food is available as well, right? So we also saw diet-dependent variations and things like that. Very simply, if you put E. coli and Saccharomyces on glucose and xylose, Saccharomyces won't use xylose, but E. coli can use xylose, right? So depending upon the other organisms that are present, there is going to be some constraint on how this organism can grow, and this hopefully tries to capture some of those things.
But all organisms are trying to maximize biomass, as Chetan was telling, right? So,
right.
I mean, in the end, if you have a finite amount of, let's say, substrates in the environment, then maybe what you say is correct. But I'm just thinking.
Yes, no. So there is a finite amount of substrate, right? So there is a given food that is given.
Okay, so you have.
So there is a EU diet or a Mediterranean diet which says there's X amount of each of the compounds.
Okay.
So yeah, maybe.
So the fraction. Okay.
Correct.
Okay. Yeah. Thanks.
But that's a good line of thinking. Yes.
So very thought-provoking talk. So I have a question related to biomass function. So biomass composition is not the same for all the organisms, right?
Yes.
And it does play a huge role in what the final objective is. So how do you account for those changing, because you're just taking these models from the database. So do you do curation on the biomass composition as well?
So this is done to an extent in the database.
Okay, so, but these are all not...
So we can't say that these are as well curated as your E. coli or even your Staph aureus or one of those, but these have been reasonably well curated, gap filled and so on, and they don't have like the same biomass objective function or anything like that. In fact, the pan model approach just adds everything and does like just an aggregate. But here every organism, and this can also use highly curated models, right? So if you have an excellent curated model of E. coli and Saccharomyces, and you want to, of course, you need more models from each of these genera to make this more meaningful. But to go back to your question, the assumption is that these biomass objective functions have been at least reasonably curated. They are not arbitrary. And I think the Agora database has, maybe Indu should add to this over coffee or something. We've looked at it carefully, and it seems that it's, at least Agora 1, 800 odd organisms, it's been fairly well curated. They have done several rounds of curation. It's not just K-based model sheet cut and paste for sure.
Thanks. The other thing I wanted to ask is, and it might be very naive, Have you used any RNA-seq data to sort of benchmark some of your flux spans, etc. in this case?
No, not yet, not yet. I mean, if we have it, we could do it, but our current assumption is that we use a ground truth, as even Chetan was asking, based on the current standard for modeling these communities, right? So where we know the exact species we are modeling. And then we throw that and see, without species knowledge, are you still able to make some narrow predictions or?
Because there is a lot of data that exists on the gut-brain axis and how the microbiome impacts it metabolically, and also with gene expression data. So that might be something you would.
Yeah, definitely. Yeah, so this would be the other end of the spectrum. So we are talking about like 16S, WGS plus transcriptomic, but definitely, yes.
So the Finnish group have a lot of this correlations.
Sure, we'll take a look. Thanks.
Okay, I can ask one question.
Yes, Arijit.
Naive question. So between the pan model and the pan-genus model that you have weighed in, so you have a weightage and weighed some of the biomass, right? In the pan-genome, I understood that, pan model, you just pool the reactions. Here,
yeah, and it's like there is a consensus biomass or something like that. There is one biomass for the entire genus in the pan model.
Yeah,
just
so between,
sort of arbitrary.
Between the two, it is clear what is the difference at the level of biomass. One is a weight sum, the other is a sort of a consensus biomass. In terms of the occurrence of reactions, what is actually, if you can simply explain us how does it differ between a pan model and a pan genus?
So, good question. So pan is everything is present.
Okay.
There's no zeros at all.
Okay.
So for us, there are zeros and ones. So some species may absolutely, there may be no Bifidobacterium adolescentis in the Bifidobacterium model at all. I could constrain it to that direction and see what it returns.
Yeah, so then what perplexes me is that you have that dendrogram plot where you see at the level of reaction occurrence they are very far apart, but then when you look at flux distribution they become very close. So can you explain at the simple way why you see that thing? You must have removed the blocked reactions when you compare it, for sure.
Yeah, see, the challenge is that there is so much variation in the fluxes, right? So I don't think we've been able to explain that yet. I don't know if Indu has a better answer later on we could discuss. But the thing is, at the reaction level, there is, in some cases, so in the 50-member community, we see the opposite behavior. In a larger community, we see the opposite behavior. And that was a little bit interesting simply because larger communities are where we expect this method to be more useful. But we probably need to look at more examples as well and figure out what's really happening there. Good point.
Okay, thank you.